So, hello, and good morning, or good afternoon, or good evening, depending on where you actually are. Just to let you know that this presentation is approved for CME credits, so there is a button down in the bottom left corner, I believe. But if you've got any questions about the CME accreditation, you can direct them to myself or Tracy, and we'll try to address those for you. So, what I'm going to go through really are a couple of case studies that describe the use of tissue microarrays as a tool for fast tracking the development of novel cancer biomarkers, and the approach that we've taken to this at our lab here in the UCD. So, and I'm specifically going to focus on biomarkers that we've developed for breast cancer, just to illustrate this. So, really, despite the fact that there's, almost 100,000 different biomarkers described in the literature for breast cancer, what's depicted on the slide here is the sum total of clinically approved biomarkers for this disease. Estrogen receptor and progesterone receptor and HER2 status are routinely monitored either by immunohistochemistry or a combination of immunohistochemistry and fluorescence in situ hybridization. There's a few cytokeratins that can be measured, and then newer multi-gene signatures like MammaPrint and Oncotype DX that are starting to change the field. But despite these sort of vast numbers of biomarkers that are described in the literature, there's very obviously a significant disconnect between our ability to discover biomarkers and our ability to actually validate those and translate those into clinically usable tests. And that's really where my lab spends a lot of its time focused. So we really start from multiple different discovery platforms, using things like sort of gene expression analysis or data from mass spectrometry or functional genomic screens in order to identify lead biomarker candidates that we can then go on and validate. And we use multiple approaches to do this. We use things like reverse phase protein arrays and antibody arrays. But what I'm going to focus on today is how we use tissue microarrays to clinically validate the biomarkers that we identify from these diverse discovery approaches, and how we actually move those one step closer to developing clinical tests for them. So, to begin with, I suppose we should define what a tissue array is, and traditionally our discovery approaches would look at large numbers of genes or large numbers of targets in small numbers of samples in order to identify those that we think are associated with the progression of the disease or the response to a drug. Whereas tissue arrays allow us to basically to invert that and to look at one gene or one protein or a small number, if we use multiplexing approaches, in tens or hundreds or thousands of cancer tissue. So it allows us to look in a high throughput manner at large numbers of patient samples so that we can determine the clinical relevance of any of the genes or proteins that we identify through our discovery approaches. So in essence, it's a very useful platform for high throughput pathology. Predominantly, tissue microarrays to date have been analyzed using things like immunohistochemistry. The vast majority of the work that we do with tissue microarrays is based on immunohistochemical detection of differentially expressed proteins. We do some work on using in situ hybridization, and I'll show you case studies for both in situ hybridization and immunohistochemistry during this presentation. And both of these approaches can be very successfully applied to examine biomarkers in large numbers of tumor samples simultaneously. Small amounts of papers have been published using approaches like infrared spectroscopy or Raman spectroscopy, and we do in the lab here do some work collaborating with groups that are involved in alternative methods of detecting protein expression signatures on tissue microarrays. And the fact that we've got large numbers of samples in highly imageable arrays allows us to look at these alternative approaches for detecting proteins expression signatures, etc. But the vast majority of the work that we do is based on immunohistochemistry or in situ hybridization. The other benefit of using large numbers of samples on single slides is that it allows us to actually scan those slides and make a digital representation of the slides. And the digital representation of the slides gives us a permanent record, first and foremost, a permanent high definition record that we can then go back to. Very often when we stain using immunohistochemistry or in situ hybridization, the stains bleach after a while. If we were to go back and scan a slide that we had stained a few years ago, certainly the staining wouldn't remain. So we get a permanent record of our slide in a digital archive, but it also allows us to then develop computer algorithms to actually aid us in the analysis of the protein expression signatures that we're looking at. And the benefits of automated analysis versus manual analysis of immunohistochemically stained tissue microarrays are manifold. Manual analysis, for example, is quite subjective and very time consuming. If you're looking at an individual case, it can be done reasonably quickly. But when you're trying to look at tens or hundreds or thousands of cores, then it gets very laborious and very time consuming to actually look down a microscope and score all of these cores individually. So it takes quite a long time. It's also very subjective in that different observers will score the same level of staining slightly differently, and that's certainly a problem if you're trying to apply a diagnostic threshold across different laboratories and at different sites. So having a reproducible data that can be generated by automated analysis certainly makes this a lot more feasible. The other major problem with manual analysis is that the human eye, while extremely complex, tends to find it very difficult to distinguish between things that are reasonably similar. So manual scoring of immunohistochemical staining tends to range from 0 through 1+, 2+, and 3+, and after that we really don't apply any more to. We may look at numbers of positive cells on top of that, but generally manual staining is scored as 0, 1+, 2+, and 3+, and that doesn't really lend itself to addressing the level of staining in a fully quantitative way. For all intents and purposes, it's mainly semi-quantitative. So and it requires a significant amount of expertise to actually examine immunohistochemical staining and interpret it properly. Whereas automated analysis is completely based on the quality of the computer algorithm that has been made to actually perform that analysis. So it's completely objective. The level of staining is determined by fine-tuning of the algorithm. It will produce reproducible data. It will apply the same algorithm to every single slide and every single section that it looks at. On top of 0, 1+, 2+, 3+, the signal is actually measured as a continuous output, depending on the numbers of colors that can actually be scanned by the scanner. So our scanner will measure basically from 0 to 300, and it will give a score anywhere within that range. So it really does open up the possibility of developing more sophisticated automated image analysis tools that allow us to objectively and reproducibly examine complex staining patterns in cancer tissues. The other nice thing about the automated analysis, obviously, is that the different colors can be separated out from each other. So in the example that is sort of playing here at the moment, the manual image is fixed, so the counter stains and the positive staining are all on the same image. But we can remove each of the different colors when we try to when we process a digital image and look at this in an automated fashion, and that allows us again to monitor the level of staining without the influence of the counter stain. So we've used these approaches over the years in the lab to develop our own automated image analysis algorithms, and then applied these to known clinical scenarios, and are now sort of have now developed sort of commercial tests based on these. So in this case, what we basically looked at was quantification of the estrogen receptor and progesterone receptor in patients that were enrolled on a clinical trial for tamoxifen versus placebo. Obviously, it was an old trial. We don't give patients placebo versus tamoxifen anymore, but this is a trial that was run many years ago to examine the effects of 2 years tamoxifen. And you can see on the slide what our original markup image was like, or sorry, what our original immunohistochemical staining image was like, and this is how the algorithm marks up that image. And then when we measure the levels of staining based on this, we can use that to help to define a threshold for ER positivity, and basically we're able to define, by comparing manual analysis to our automated analysis, we see a very strong correlation between where we would class the 10% cutoff for ER positivity based on the automated staining compared to how 3 independent pathologists would score. In fact, the level of concordance between so the replicates of our algorithm is actually better than the level of concordance between 3 independent pathologists. And this, to a certain extent, is a significant problem in ER staining across laboratories, even though this is a standard immunohistochemical stain that's performed on practically every breast cancer patient that walks into a clinic. There's still a level of discordance between the determination of ER positivity across different labs in different countries, and this is you know, a diagnosis that's used to determine whether or not patients should actually get hormone therapy or anti-hormone therapy. So fine-tuning how ER positivity is measured is actually pretty important. But we can see quite nicely that the algorithm is able to pick out ER positive patients, and then when we compare those who are treated to those who are untreated with tamoxifen, there's obviously a clear benefit to treating patients who have ER positive, who are ER positive with tamoxifen. So this has led to the development of a proprietary automated image analysis tool called IHC-MARK that is being developed and marketed by a spin-out company from UCD here called Oncology Limited, that's led by Professor William Gallagher. And this approach basically can identify membranous staining, or cytoplasmic staining, or nuclear staining, and will produce digital markups for all of these, allowing us to do automated analysis of the levels of expression of these. So we've used this approach, for example, to look at the expression of MSMB in over 3,000 prostate cancer samples, and again we can see the normal manual analysis image, and then our markup digital image that's been analyzed by the IHC-MARK algorithm, and this again allows us to track what the levels of expression are like. When we couple this with the clinical data from these patients, it allows us to see that patients with high expression of this biomarker do much better than patients that have low expression of this biomarker. And again, as I said, this has been validated in over 3,000 patients. So the algorithm sort of works very well for distinguishing cytoplasmic, membranous, or nuclear staining and quantifying that, allowing us to get a continuous output to do sort of rigorous statistical analysis of the correlation of expression with outcome in these patients. The other nice thing, I suppose, that automated image analysis allows us to do is to look in a little bit more depth at what's actually happening within the cancer tissues. And one application that we've used our algorithm for is to quantify the level of infiltrating T cells in breast tissues and correlate that with outcome in patients. So in this case, this was a collaboration with David DiNardo and Lisa Cousin's group, and what David had been looking at was the impact of inflammation basically on breast tumor metastasis. So he had MMTV, I think there were MMTV polyoma middle T mice that generated basically generated breast cancer, transgenic mice that generated breast cancer, and then spontaneously would metastasize to the lungs. But when he crossed these mice with mice that either had a RAG1 knockout, so they were deficient in VDJ recombination and therefore couldn't make any new antibodies, or crossed them with mice that were deficient in CD4 or CD8 cells, he saw absolutely no metastasis from the primary tumors to the lungs whatsoever. The interesting thing is the primary tumor burden doesn't change in these crosses. So the primary tumor is exactly the same, but removing the effect of inflammatory cells completely impacts on whether or not these breast tumors move from the breast and move to the lungs. And considering that the vast majority of patients who die from cancer die from metastatic disease, being able to modulate the level of metastasis is obviously something that was very important. So David had already done these mouse experiments when he came to us and basically asked if we could help him to look in breast cancer patients to see whether or not the levels of infiltrating T cells in the tumor were could actually correlate with the outcome of these patients. So we used a combination of our IHC-MARK algorithm and a pattern recognition algorithm to delineate whereabouts in the tumor or whereabouts in the surrounding tissue the infiltrating T cells actually were. So this was our original IHC image. Using a pattern recognition algorithm, we were able to determine whether or not there's which parts were tumor, which parts are stroma, and then staining for either CD8 or CD4 or CD68 T cells. We were able to mark up the actual levels of the T cells, and then using our pattern recognition algorithm determine what the level of T cell infiltrate into the tumors were, and then correlating that with clinical outcome, determine whether it had any impact on how the patients actually did. And we saw quite nicely when we looked at this that the levels of CD8 positive T cells in conjunction with the levels of CD4 and CD68 T cells in these patients completely predicts how the patients will actually do. And this tied in very nicely with the work that David had done, showing that having a functioning inflammatory system was crucial to the metastasis of breast cancer from the original primary site to the lungs. So to do all of this work, really, your discovery efforts tend to find all sorts of putative biomarkers. Some of them are sort of easily commercially available. Lots of the biomarkers that you identify are for novel proteins or novel genes or things that really we don't have proper tools for doing immunohistochemistry yet. So in order to address that, we really partnered with a major project that's run from Uppsala University in Sweden called the Human Protein Atlas, and they've got a website, proteinatlas.org, that is aimed at firstly generating antibodies to all known human proteins and then mapping their expression in both normal human tissues and cancer tissues, as well as cancer cell lines by immunocytochemistry. So we've partnered with the Protein Atlas over a number of years to develop novel antibodies to biomarkers that we have identified through our various different through our various different discovery platforms, and then taken those novel antibodies and brought them into our tissue microarrays in order to look to see what effect the expression of our biomarkers is having on the outcome of the patients. So what the Human Protein Atlas actually do is to design what's known as a protein expression sequence tag that is a small peptide that's unique for the protein of interest to allow the generation of a monospecific antibody that can be then used to detect it. And then they go through a series of steps of purifying the prests, expressing the prests, generating antibodies, and then purifying these monospecific antibodies. The monospecific antibodies at this point, these are rabbit polyclonal antibodies that are monospecific rather than monoclonal antibodies, and these are just then tested briefly by Western blot and predominantly also by a protein array. So all of the different prests that are generated are also spotted on these protein chips and are used to test the specificity of the antibodies that are actually made. So we submitted, a number of different putative novel biomarkers to the Protein Atlas in order to develop new antibodies to these. Some of them did have commercial antibodies available, but when we tested them for immunohistochemistry, they were insufficient. They don't really work. I mean, this is a problem that we encounter regularly when we look at commercial antibodies. It's a problem that actually probably even exists within some diagnostic antibodies, in that there's quite a large amount of non-specific binding of these antibodies, and we put all of our antibodies through sort of a rigorous sort of testing before they go near any of our our tissue microarrays. So we submitted a number of biomarker candidates to the Human Protein Atlas, and they very kindly generated about 200 different antibodies to novel proteins that we then moved on to validate in our tissue microarray bank. So what we do when we get our biomarker is, we've used whatever biomarker discovery approach we think is suitable. So we use gene expression profiling using DNA microarrays. We do next generation sequencing using RNA-seq. We do various different proteomics-based discovery approaches and lots of other genomics-based approaches. But once we've identified our biomarkers, the Human Protein Atlas will make us an antibody, and then we move through a series of steps where we validate the specificity of that antibody and its probable utility for immunohistochemistry. So our first step really is just to check what the specificity is like by Western blot analysis, and we would expect to see a single band on a Western blot for any antibody that we then progress to use for our tissue microarrays. Our next step really then is to very often at this second step we very often also perform QRT-PCR analysis to get an example of what the level of expression of our biomarker is. But both Western blot analysis and QRT-PCR analysis alone are really insufficient for determining the level of specificity of the antibody. What you really need to do is to check how specific the antibody is in the same chemistry that you're actually going to use it for your diagnostic test. And in this case, we're performing immunohistochemistry, so we evaluate the specificity of our antibodies using cell pellet arrays. And cell pellet arrays are basically plugs of cells that we formalin fix and embed in paraffin and process in exactly the same way that we would process tissue that we then use for our tissue microarrays. So the chemistry is the same. They're both formalin fixed, paraffin embedded, but in this case we've got a selection of different cell types, and the simplest thing to do really is to take cells that are negative and positive for the biomarker that you are trying to detect. If there aren't cells that are negative and positive, what we do is we tend to modulate the expression of our target biomarker, either using shRNAs to suppress the expression of the biomarker or using cDNA expression to express the biomarker. So using these cell pellet arrays, we're able to determine the level of specificity of our antibodies. Really what we want to see is a signal in the cell lines that express the protein and no signal in the ones where we've either knocked it down or where they don't express it. We then move on to assess the specificity usually on full-face tissue sections, actually, again where we've already performed QRT-PCR analysis and we know from the same tissue, so we know what the gene expression is like. and we look at our level of specificity on that, and then we would assess our biomarker using, first of all, a screening array of about 100 different tissues, and then it would finally, if it's passed all of these steps, would move on to use on our full tissue microarray. Once we put it onto our full tissue microarrays, then we can carry on to do our automated image analysis, correlate with our clinical data, and use this really as a way of moving candidate biomarkers through large numbers of clinical samples in order to determine whether or not they have any clinical relevance at all. So what I'm going to do really for the rest of the talk is to concentrate on two particular case studies that addressed two clinical problems that we thought were relevant, and that we could use our tissue microarray program here at UCD to examine. So in the first case, really, what we're looking at is the problem of whether or not patients with early stage lymph node negative, hormone receptor positive, HER2 negative breast cancer require chemotherapy, or if they get anti-hormone therapy alone, will they actually respond to that? And really what this boils down to is a question that has sort of taxed clinicians for a number of years now, and this really is whether are we actually over-treating node-negative breast cancer patients. Recent data would suggest that somewhere in the region of 70% of early-stage node-negative breast cancer patients will do just fine with surgery, radiotherapy maybe, and anti-hormone treatment alone, and certainly don't need cytotoxic chemotherapy on top of that. So the problem with not over-treating patients is that we end up under-treating them. So in order to accurately determine the provision of therapy, we really need unambiguous biomarkers that can determine the need for adjuvant chemotherapy in these patients. And this is certainly something that Laura Van Veer and colleagues were well aware of back in about 2000 when they set out to examine this exact problem. And what Laura and her team did really was to look at about, in the first instance, 78 node-negative breast cancer patients and profile all of the genes that were differentially expressed in these patients. And she published this original paper back in 2000 in Nature, where they described a 70 gene signature that could predict outcome in 63 out of 78 node-negative patients. This 70 gene signature was then further validated by Mark van de Vijver and Laura and other colleagues, and published in the New England Journal of Medicine, and is now commercially available as the sort of MammaPrint test, which is under validation in the MIND-ACT trial. And basically what this test does is measures these 70 genes and determines those patients who are at high risk. So those patients get chemotherapy, those patients who are at low risk, and those patients don't get chemotherapy. And the discordant cases within the MIND-ACT trial have been randomized to receive either chemotherapy or no chemotherapy. Oncotype DX and the TAILOR-X trial have worked pretty similarly, where instead of using 70 genes, Oncotype DX measures 16 genes and 5 control genes by a QRT-PCR assay, and again determines the need for adjuvant chemotherapy in early stage lymph node negative breast cancer patients. So patients with a high Oncotype DX recurrence score should get chemo. Patients with a low Oncotype DX recurrence score are spared chemotherapy, and patients that have an intermediate score within the TAILOR-X trial were randomized to receive either chemotherapy or no chemotherapy. And we should find out by ASCO next year how what the first report of that trial is like. The major problem with both tests really is that the intermediate patients are patients that we really don't know what to do with. And within the TAILOR-X trial, this was at least half of the patients, probably up as high as 60% of the patients had an intermediate score. So we really don't know whether to give those patients adjuvant chemotherapy or whether they're safe on hormone therapy alone. So those are exactly the types of patients that we need to find new biomarkers for, to add to MammaPrint or Oncotype DX testing, and to add to the normal testing that we actually do with these patients to try to discover what the most appropriate form of therapy for them is. So we worked with Laura and her colleagues to re-examine the data set that they actually developed the MammaPrint test from in the first place. So we sent a bioinformatician over to Agendia who spent 6 months reanalyzing the data over there, using different approaches to those that were used for the original study. And we basically defined a list of 100 transcripts that were associated with good prognosis and 100 transcripts that were associated with poor prognosis. Rather reassuringly, in a completely unbiased and unsupervised fashion, markers that are sort of already associated with good prognosis, like estrogen receptor and progesterone receptor, show up quite clearly on the side of the top 100 good prognosis genes. And over the ensuing years, we published a number of papers on other markers from the poor prognosis side, and describe their association with breast cancer. So the case study that I'm really going to sort of show today is the example of a protein called CART, or the cocaine and amphetamine regulated transcript. So from this reanalysis, for example, we identified CART as being in the top 5 of our poor prognosis genes. And CART peptides are reasonably interesting. They're neuropeptide hormones and neurotransmitters that are produced in the arcuate nucleus of the hypothalamus, and they act as anorectic neuropeptides that control feeding, body weight, and a number of endocrine functions. Their structure is pretty much conserved across species, so human, rat, and mouse active CART peptides are 100% identical. There's absolutely no change in the protein structure for them. In terms of cancer, circulating CART levels have been demonstrated in neuroendocrine malignancies, but there have been no previous link between CART and breast cancer. It should be noted that CART isn't the only neuropeptide to be implicated in breast cancer. Neuropeptide Y, nerve growth factor, vasopressin, oxytocin. There's a number of other neuropeptides that have been shown to play a role in breast cancer. But we had seen, absolute gene expression in breast cancer rather than sort of hypothalamic CART having an impact on breast cancer. But we had actually seen CART being expressed in breast cancer tissues, so we wondered what role it might actually play. So we first went and looked at whether or not CART was expressed in normal breast tissue, and found that no matter how hard we looked, we couldn't see CART expressed in normal breast tissue at all. But we could see CART expressed in primary breast tumors. And whenever it was expressed in primary breast tumors, it was also expressed in corresponding lymph node metastases from these same patients. So we looked, in order to do this, basically we used patient chips that had normal breast tissue, primary tissue, and metastatic samples from 12 different patients. And in about 15% of cases we saw positive samples in the primary tumor. And as we said, whenever it was positive in the primary tumor, it was also always positive in the corresponding lymph nodes from these patients. So we developed both a manual and an automated scoring model, sort of manual scoring model and an automated scoring model, in order to measure CART activity or sort of CART expression in breast cancer, and saw a very good correspondence between our manual scores and our automated scores, with a very high R number of about, or squared of about 0.79. So we, using this, defined basically a cutoff of 3+ for high CART, and 0, 1+, 2+ for low CART, or we had an RGB score that we applied across multiple cohorts to actually define high CART versus low CART, and therefore be able to examine its expression and correlate that with outcome of these patients. So to do this, we looked at 2 different tissue microarrays. The first one is a cohort of 512 consecutive breast cancer cases that were that were identified in Malmö University Hospital in Sweden. These patients were predominantly postmenopausal, with a median age of 65 years. And we had a median follow-up on these patients of about 11 years. The second cohort was was actually the breast cancer cohort that I described previously. So these were premenopausal women with primary breast cancer who were enrolled in a randomized trial and assigned to either 2 years of adjuvant tamoxifen or placebo. And we looked at the impact of CART on outcome in these two different breast cancer cohorts. So in cohort 1, using our tissue microarrays and our automated image analysis, we were able to show that high CART expression correlated with a poor outcome for these patients. In this case, we're looking at overall survival. We see pretty much the same effect when we look at breast cancer specific survival. If you look at recurrence-free survival, we see the same effect, but here we've shown overall survival. And patients that had high CART did worse basically than patients that had low CART expression. If we specifically look at what we're really interested in, were these lymph node negative patients, and trying to determine whether or not they would respond to tamoxifen or whether they would actually need additional chemotherapy. When we specifically looked at lymph node negative patients, again we see this same pattern where patients that had high CART did a lot worse than patients that did low CART. So the conclusion that we drew at this point was that high CART expression correlated with poor outcome in postmenopausal patients, and this remains the case in lymph node negative postmenopausal patients. When we looked at the second cohort, these are the patients from the randomized trial, and these were premenopausal patients. In the cohort as a whole, CART levels had no impact really on how the patients actually did. But when we focused in again on lymph node negative tumours, there was a very clear effect of high CART predicting poorer outcome in these patients. And there's a lot of what are generally called prognostic biomarkers described in the literature. Truly prognostic biomarkers describe what the natural history of the disease will be if the patients are left untreated, if they're not actually treated. The vast majority of prognostic biomarkers that are described in the literature are described in retrospective cohorts where patients have actually received some form of treatment, so therefore they're not actually truly prognostic. But this cohort actually did have half of the patients who were randomized to receive placebo and received no treatment. So we were able to look not only just at lymph node negative tumours alone, but we were able to look specifically at untreated lymph node negative tumours and examine the pure prognostic potential of CART in these patients. And again, we saw a very clear difference between patients that had low CART and patients that had high CART. So those patients that have high CART and had untreated lymph node negative tumours did far worse than those patients that had low CART. So and for this cohort and for the previous cohort, we performed a multivariate Cox regression analysis and looked at controlling for age, grade, tumor size, ER status, HER2 status, Ki67, and in all cases, CART is an independent prognostic indicator in both of these cohorts, both in postmenopausal patients across the board, but in premenopausal patients, in lymph node negative premenopausal patients, and certainly in this case with a hazard ratio of 4.81 for high CART expression compared to patients that had low CART expression. So our normal sort of modus operandi when we find something like this is try to pick it apart and see how it actually works. So we looked at some breast cancer cell lines to see whether CART was actually expressed, and as with all best laid plans, we found that CART isn't expressed at all in any of the breast cancer cell lines that we have. We do have some rat hypothalamus as a positive control, so our experiment works. It's just not expressed under normal circumstances in breast cancer cell lines. At this point, we thought maybe it was because it was under under some kind of endocrine control. And indeed, all of the sort of endocrine-driven tumours that we'd looked at, for example, endometrial cancer or ovarian cancer or prostate cancer, we could see CART-positive cases in all of these, whereas a lot of the non-endocrine-driven cancers, we didn't see much in the way of CART expression. So if we can't switch it, and normally what we would do is we would find a cell line that expressed lots of CART, switch it off using shRNAs and find out what effect it has on the cells. So if we can't switch it off, our next port of call really is to switch it on. So we took ER-positive breast cancer cell lines, MCF7 cells, and we basically cloned CART, and we transfected it into the cells compared to untransfected cells. And one of the first things we noticed was some degree of stabilization of the estrogen receptor in these cells. We also saw in the cells where we had re-expressed CART an increase in the levels of progesterone receptor. And at this point, really one of the only things that was known about CART signaling was that it binds onto an unknown G protein coupled receptor, and that it activates the MAP kinase pathway. So we should see MEK activation and ERK activation if CART is actually having the same effect in breast cells as it has in other ones. So we looked and we saw again that when we express CART in breast cancer cells, we see an increase in the amount of ERK phosphorylation, and this suggested that it was still activating this pathway. And CART is also a secreted peptide, so we also wondered if we expressed CART from inside breast cancer cells, would it be processed and secreted back out? So we looked in the tissue culture media, and we could see secretion of CART in these cells. So we wondered, well, if we take this conditioned culture medium that has CART in it and put it back on cells that don't have any CART in them, what effect would it have? And what we found was that this was the way that we could actually get CART to be expressed. So in cells that are treated with CART conditioned medium that don't have any, we haven't cloned any CART into them, all of a sudden CART expression gets switched back on. And we can measure this by Western blot, and we can also measure it by real-time PCR. So it is really activating the transcription of the CART gene. So what we postulate here really is that we end up with an autocrine loop where CART amplifies its own expression and its own signal. And this certainly makes sense when we look back at the tumours, and that when we see cells within the tumour that are positive for CART, all of the cells tend to be positive for CART. So we think that when the tumour expresses CART, it induces the expression of more CART within the cells. And this happens in multiple cell types, so it doesn't really depend on, these are MDA, MB231 cells that don't have any estrogen receptor. These are MCF10A cells that are, these ones here are MCF10A cells, so these are almost normal cells. So we don't really see much in the way of an impact of the estrogen receptor on this signaling cascade. However, the fact that we had sort of noticed, a small increase in the amount of estrogen receptor, but definitely an increase in the expression of ER target genes, led us to wonder whether CART was having any particular effect on the transcriptional activity of the estrogen receptor. So we basically treated cells with, the nice thing about CART actually, is that on top of the fact that it gets secreted into culture media like this, so we can collect conditioned culture media. You can also buy recombinant CART peptide, which allows us to perform these experiments just using recombinant CART, and makes it a lot cleaner. So we treated MCF7 breast cancer cells with recombinant CART peptide, and saw at least some stabilization of the estrogen receptor. We saw this, as we expected, this increase in the levels of phospho-ERK after we treated with recombinant CART. And there's multiple different ways in which the estrogen receptor can actually get activated. Normally, estradiol binds onto the estrogen receptor, and this causes dimerization of the estrogen receptor, binding to an estrogen responsive element, and transcription of ER responsive genes. However, growth factor binding to growth factor receptors can activate the MAP kinase pathway, and this MAP kinase activation can lead to phosphorylation of the estrogen receptor on a different site at serine 118, and this can still lead to activation of ER-driven transcription and expression of ER target genes. So given that we had already seen that CART could activate the MAP kinase pathway, we wondered whether or not it actually led to the phosphorylation of the estrogen receptor at another site. So we looked at the expression, or looked at phosphoserine 118 levels, and indeed found that CART activates phosphorylation of the estrogen receptor at serine 118. And this phosphorylation event is normally a transcriptionally activating event on the estrogen receptor. So we examined what effect that had using an ERE luciferase reporter assay to measure estrogen receptor transcription activity, and certainly found that CART was able to activate an ERE luciferase reporter almost as efficiently as estradiol itself. And indeed, in addition to that, it also switches on the expression of the endogenous ER target gene, progesterone receptor. If we block the MAP kinase pathway using a MEK inhibitor, so we block the phosphorylation of ERK, we lose this stabilization of the estrogen receptor, we lose the accumulation of estrogen receptor phosphorylated at serine 118, and we no longer get this induction of ER target genes. Indeed, if we mutate serine 118, so this is wild type estrogen receptor versus wild type CART, and it can switch on the ERE luciferase reporter, whereas when we mutate the serine 118, it blocks the ability of CART to activate this ERE luciferase reporter assay, whereas estradiol, which stimulates phosphorylation at different sites, doesn't block, so mutation of serine 118 doesn't block that ability. So certainly what we think we've shown at this point is that CART binds to this unknown receptor, activates RAS, so we can see when we treat with CART compared to a positive control, we see this very strong activation of RAS, which activates the MAP kinase pathway as evidenced by phosphorylation of ERK. This leads to phosphoserine 118 estrogen receptor phosphorylation, which leads to the transcriptional activation of the estrogen receptor and a tissue response, which we can measure based on the expression of ER target genes. So what does this actually sort of mean, and what effect does it have? Well, these signaling pathways and mechanisms of activation of the estrogen receptor have an impact on how the estrogen receptor responds to tamoxifen. So tamoxifen normally binds at the estrogen receptor, blocks the phosphorylation of it, but it doesn't block phosphorylation at serine 118. So this is a method of activating the estrogen receptor where that should be able to circumvent the effects of tamoxifen. So if we go back in and look in our breast cancer patients and look at the effects of CART, certainly those patients that have high CART have high expression of ER target genes. When we look at the loading of the estrogen receptor onto the promoters of these genes by chromatin precipitation, those that are treated with CART have the estrogen receptor on their promoters. And when we express CART in cells and then treat them with tamoxifen, certainly those cells that have high CART compared to the control cells or the tamoxifen treated alone certainly have much better protection against tamoxifen-mediated cell death compared to those cells that have low CART. So we went back into our breast cancer cohorts and looked at what effect CART had by tissue microarray analysis on the survival of patients who've been treated with tamoxifen. So specifically looking at postmenopausal patients who had received tamoxifen, and again found that patients that had low CART did much better than patients that had high CART, suggesting that high CART correlates with poor outcome in ER positive postmenopausal patients who've been treated with tamoxifen. When we looked at patients from the randomized trial and stratified them according to CART levels, patients that had low CART had a significant response to tamoxifen compared to control, whereas patients that had high CART had a non-significant response, even though there is still some separation of the Kaplan-Meiers. Patients within this cohort only got 2 years of tamoxifen, whereas current standard of care is at least 5 years, and recent data suggests that patients should probably be getting 10 years tamoxifen treatment. And when the data is presented from 10 years tamoxifen treatment and 15 years tamoxifen, it may suggest that even longer treatment with tamoxifen gives better results. So we looked in a third cohort and looked at the effects of CART on outcome in these patients, and again saw when looking at both recurrence-free survival and overall survival that patients that have high CART do much worse than patients that have low CART, and again all of these patients were treated with tamoxifen. And again, both in this cohort and in all of our other cohorts, the multivariate analysis controlling for age, grade, stage, tumor size, ER status, PR status, HER2 status, Ki67 status, confirms CART as an independent prognostic factor in hormone receptor positive tamoxifen treated patients, and suggests that patients that have high CART really don't do very well on hormone treatment, and would be candidates for receiving more aggressive chemotherapy added to their regimes. So at this point, what we've done is looked at CART in over 1,000 breast cancer patients using tissue microarrays, and developed what we would suggest is sort of fairly compelling evidence that CART is an independent prognostic factor in lymph node negative breast cancer. So currently what we're doing is we're examining CART levels in intermediate patients from the TAILOR-X trial, so patients who have a discordant diagnosis where we don't know whether they need to get chemo or whether hormone therapy alone is sufficient. So they've been randomized to get one or the other. And we're looking at CART in those patients to see whether it can help to determine whether they need to get chemo or don't need to get chemo. We've also shown what we think the mechanism for this poor response to anti-hormone therapy is, and this is that we see a ligand-independent activation of the estrogen receptor, and that this leads to poor response to anti-hormone treatment in both pre and postmenopausal patients. Another thing that would be really important for us to look at is the effect of aromatase inhibitors in postmenopausal patients. But given the mechanism of action of CART and the fact that it's a ligand-independent activation of the estrogen receptor, we think that aromatase inhibitors will have very little effect on the activity of CART in these patients. So that's an example of how we've taken a biomarker that we discovered through bioinformatic reanalysis, used tissue microarrays to clinically validate it in large numbers of patients, and then I suppose there's a trend towards using multi-marker panels. And less support for individual biomarkers these days, which is one of the reasons why we try to perform this mechanistic anchoring, in that I think there's probably safety in numbers when we have no idea what the individual parts of our signature actually do at a biological level, or how they might contribute to disease progression. Whereas when we have individual biomarkers, if we can explain how they actually work, that mechanistic anchoring gives us a little bit more sort of confidence in their possible use. So then we can just objectively look at what impact CART has on outcome in as many breast cancer patients as we can, and especially in these discordant TAILOR-X patients. So that's using sort of immunohistochemistry, automated image analysis, and large numbers of patients to sort of validate biomarkers in that way. The other approach that we've used is to look at microarrays and to look at in situ hybridization as a way of detecting biomarker expression in breast cancer. So I've only got a few minutes left, so I'm going to run through this part of it a bit more quickly. But basically we took two different clinical cohorts where we had gene expression signatures, gene expression profiles and outcome, and used a bioinformatic approach called co-inertia analysis to identify which microRNAs might be associated with outcome in these patients. So basically we had gene expression data for all of these patients, and we had whether or not these were good responders or poor responders across the top. And then using a selection of different microRNA binding site databases, we were able to also to plot all of the microRNA binding sites and infer which genes are regulated by which microRNAs. So we end up with two linked plots, with a top basically and a bottom, and on the top the all of the poor prognosis and good prognosis samples clustered together, and at the bottom divided by their gene expression analysis and the inferred microRNAs that might regulate those. We're able to identify microRNAs that contribute to good prognosis and microRNAs that contribute to poor prognosis. Obviously this is a cartoon, so things never separate out as perfectly as that in real life, but when we looked at two independent data sets where we had outcome and gene expression data, we were able to separate out samples that patients that had poor prognosis from patients that had good prognosis, and a microRNA miR187 that was associated with poor prognosis in these patients. We saw exactly the same thing when we looked at a second independent cohort. In this case, the poor prognosis samples are on the bottom and the good prognosis samples are on the top. But again, miR187 turned out to be the microRNA most associated with poor outcome in these patients. So again, our approach is to modulate the expression of this in breast cancer cell lines. So we found a breast cancer cell line, for example, MCF7 that doesn't express miR187, and we use a lentiviral expression system to express the microRNA in these cells. We also picked a second cell line, SKBR3, and expressed it in those, and then looked phenotypically at what effect it has. So when we switch on miR187 in these cells, it makes both MCF7 cells and SKBR3 cells grow more and be more tumorigenic. It affects their migration and it makes them close a wound faster, it makes them more migratory. And if we look at the level of invasiveness using an invasion into collagen test, we can see that the cells that have the microRNA expressed really invade into the collagen matrix, whereas the cells that have the vector or control don't. So stable expression of this microRNA in breast cancer cells makes them more invasive, makes them more migratory, and makes them more aggressive and more tumorigenic. So again, we've got a phenotype that's associated with the sort of overexpression of this microRNA. We went back into our tissue microarray cohorts and we're able to look using chromogenic immune in situ hybridization at what the levels of expression of this microRNA were and correlate that with clinical outcome. So again, we use these double deoxygenin or DIG labeled locked nucleic acid probes to measure the microRNA expression in the tissue. So we're able to plot no expression, expression of so 1+, 2+, and a 3+ expression, and then compare that to a scramble probe in order to measure the levels of this microRNA in our tissue. We also took QRT-PCR, real-time PCR samples from some of these cores and correlated the in situ hybridization score that we saw with the QRT-PCR score, and again, all of those samples that were deemed low expressors or negative expressors by QRT-PCR were exactly the same ones that we picked up by in situ hybridization. So we were quite happy that we could accurately measure this. And basically, what we found using this was that across our first cohort, that there was a non-significant association with outcome in all patients, but in lymph node positive patients that had high expression of this microRNA had a significantly poorer outcome than patients that had low expression of this microRNA. Again, in our second sort of validation cohort, we had more patients. So across all patients, the high expression of the microRNA correlates with poor outcome in these patients, but very strongly high expression of the microRNA correlates with poor outcome in lymph node positive patients compared to lymph node negative patients. And again, patients that have 1 or 2 positive lymph nodes but don't yet necessarily have metastatic disease, we really do need to find biomarkers that can help us guide therapy in these patients and suggest which patients again would be could be treated with adjuvant chemotherapy. So this was looking at breast cancer specific survival. If we look at if we look at recurrence-free survival, we see exactly the same thing again, that the lymph node positive patients that have high expression of this microRNA do really poorly compared to patients that have low expression of it. And again, if we using multivariate models correcting for age, tumor size, grade, ER status, PR status, HER2 status, these microRNAs at miR-187 is an independent predictor of outcome in these patients, and especially in lymph node positive patients compared to lymph node negative patients. In each of these cohorts, lymph node negative patients, miR-187 expression doesn't have any impact on their outcome. So what we really have sort of shown here in this part is that we can use tissue microarrays not only to look at protein expression by immunohistochemistry, but also use in situ hybridization in a high throughput fashion to detect microRNA expression, and this can be done for messenger RNA expression as well, and then also use this as a way of correlating the expression of specific microRNAs within the tissues themselves with outcome of these patients. And this gives us, significant benefits over using things like real-time PCR-based assays for microRNAs, where tissue architecture isn't maintained and all of the RNA from the whole tumor is removed. So if that's the approach that you use, you really don't know whether the microRNA is expressed in the stroma or whether it's expressed in the tumor. But actually looking at it using a tissue microarray allows that tissue architecture to be preserved, and it allows us to specifically measure tumor expression of the microRNA and correlate that with outcome of these patients. So anyway, I hope that has given you some insight into the kind of work that we do here in UCD and how we've used tissue microarrays to examine large amounts of cancer samples and validate our biomarkers using those. Okay, great. Thank you.